kind words and also allow me to have the final talk of the day.
Yes.
Big honor to be in front of this expert crowd, obviously. And my presentation will also be a little bit more in line with what Manus Donahue just also showed, which is very exciting to also use it as a clinical application. How Bolt CPR has been done for decades now in Toronto. Utrecht has also very successfully translated it kind of like a routine workup for certain patients. It's exactly also the efforts I have tried to reach, or at least to accomplish I started in 2013 as a resident, and then also started building the labs over the past decades. It has taken a decade, to be honest, but to really get Bolt CPR so much in shape that we can really say, well, we have an experimental advanced hemodynamic imaging technique that can for clinical purposes. And obviously, when you start, so again, 10 years ago, if you want to convince clinical neuroradiologists, clinical MR technicians with 2 big gas bottles and a respiratory device, and the fear in their eyes, I will never forget. You don't think it would explode or at least be drawn into the MR. So this is also the practical implications, right? You have a cool technique, or at least on a methodological, preclinical level, you believe in a certain technique, try to optimize the methodology, bring it into a clinical practice is a whole different beast. So again, compliments also to the other groups to have achieved that. So we stick to a very simple protocol of really Bolt. We only use Bolt contrast with 1 CO2 stimulus given by the RESPIRACT. I elaborate a little bit more on that. We use the same color scheme for clinical interpretation. So blue is bad. This would be like severely impaired dynamic failure in the right hemisphere. And my main interest is really hemodynamic impairment in that regard. So, and specifically for this talk, I would also like to talk a bit about stroke risk. Not the MOIA population, that's again a whole different patient group. It's really atherosclerotic disease, which I will elaborate a bit more on today, including the ischemic stroke. But before I start, I would also use this platform to also acknowledge the pioneers in this work. And Joe Fisher is in the crowd. Together with Dave Michaelis, they really started this whole principle. Even it was unthinkable for 20 years to even start using Bolt for these purposes, and to be able to also develop this respiratory device that you can really target CO2 and also target O2 in that regard. But really have this kind of advancements already 20 years ago really boosted the field, different universities, and definitely made our work in Zurich possible. And kind of show the journey. This is a very nice review that Joe and David published 2 years ago. This really nicely also kind of put us in the context of how the development has been. We bring hemodynamic imaging or cerebrovascular reactivity imaging in particular into definitely a nice review to read. So how I got into this whole field, I was actually appointed at neurosurgery as a PhD candidate, I think about 15 years ago or 12 years ago. And that was actually with the clinical, it was actually already the plan that time to really scan MOIA patients. So basically same as the Vanderbilt group, really look at hemodynamic failure in these ischemic MOIA patients that were symptomatic and needed potentially a bypass flow augmentation surgery. And in order to make this happen, I was then integrated in the group of Joe and David to really use Bolt CPR for this specific purpose. So we already started working on Bolt CPR. This whole effort also make as a neurosurgeon today, still I'm interested in imaging research, which may seem an odd combination at first. So the outline for today's talk specifically, why would we even measure CPR? Why would we be using the Bolt contrast? I mean, it's also not an obvious one, at least not 10 years ago, maybe a bit more now. And then specifically 2 projects I would like to highlight to ischemic stroke patients. So determine the risk for recurrent ischemic events. So we have a lot of patients coming into practice that have had a stroke, become newly symptomatic again. And then it's very hard to differentiate which patients have hemodynamic impairment or actually are at high risk for recurrent stroke. So atherosclerotic patients with large vessel disease, I have to say. So this is where we try to apply Bolt CPR. And kind of as a new development in the last couple of years, we also look at patients with acute ischemic stroke. So presenting with ischemia, coming to the clinic, and then after we've done the clinical workup, meaning CT perfusion, endovascular thrombectomy, yes or no, we do a Bolt CPR within 72 hours in those patients that remain symptomatic and look at this kind of like new concept that has arisen the last couple of years, this reperfusion failure, meaning thrombectomy was successful, so the vessel was opened again acutely. However, the patient is still symptomatic and shows hemodynamic impairment. So this is a very interesting field of research. So these two things I'd like to highlight a bit. So if we look at potential imaging parameters for hemodynamic impairment, it's a bit of repetition for most, obviously, but I think for the flow, good to understand how it also historically went, because at least I, in my PhD time, grew up with a lot of the OEF, the PET studies to really look either at OEF, which would be in the middle, look at hemodynamic failure. So hemodynamic failure stage 1 would be baseline perfusion would be normal. If you give a vasoactive stimulus, the hemodynamic impairment would show. Stage 2 would be including hemodynamic impairment already at baseline. If you do then a vasoactive stimulus, you have a worse hemodynamic failure. Stage 3 would basically already be ischemia, so not to confuse stage 3 with another hemodynamic impairment group. It's really stage 1, stage 2, which are of interest, and in particular stage 2 obviously would be those patients at highest risk for ischemic events. So what we know with OEF is that we measure with PET, OEF is increased. That's what we learned. There's certain cutoffs for that. Doesn't seem to work always, maybe even biphasic. So most severe patients we even show a decrease in OEF. So OEF is a single parameter people get more and more skeptical about. CBF obviously can also be measured eloquently with water PETs, ASL as an emerging MRI technique. For stage 1 may be normal for baseline, so you really need to apply a vasoactive stimulus to identify those patients that may then show hemodynamic impairment, which is also not that easy in bilateral disease, we just heard. What I like about CVR, cerebrovascular reactivity, is that it kind of gradually decreases. So already in stage 1, you can already find impaired CVR. It may not be higher stroke risk per se, but at least you can kind of track what would be impaired CVR all the way to severely impaired, still phenomenon that way, towards ischemia. So this is a parameter we chose to pick. Every expert group has its own liking of which imaging modality he or she uses and which vasoactive stimulus. We stick to CVR. And what we know is that value in patients with atherosclerotic cerebrovascular disease is that the overall risk for these patients, so patients with large vessel atherosclerotic disease, the overall stroke risk would be 10 to 22% if you look at the historic and current literature. It's interesting because if you then take the extra parameter of hemodynamic impairments into account on top of this, the risk increases to 44%, so twofold. So it's more and more recognized that we also need a certain imaging modality to really assess hemodynamic impairment in this patient group with stroke. Some classic literature I highlighted, and these are different modalities, but kind of show the same stroke incidence. And again, with hemodynamic impairment, the stroke increase is a very important consideration. I'll get back to that. So how to measure it? We've heard different techniques. I think all techniques have pros and cons. I think transcranial Doppler is very robust, obviously, for bedside investigation. You can use it with vasoactive stimulus, obviously. It doesn't show you brain tissue perfusion, so that's why I think we all tend to do more of an MR or PET-based technique. PET for clinical purposes, as we heard already before, it's very hard, very expensive. You cannot really use it as a routine technique. MRI, I think, has the most potential. However, only little groups, they make it into the clinical translation to really make it a integration into your clinical protocol. Vasoactive stimulus, Diamox, I mean, again, this is whatever you like to do. We decided not to do Diamox because you need a baseline scan, you need to inject the Diamox, wait a certain amount of time and do the second scan. It's quite a long acquisition period. With CO2, at least, you can use it in one scan. It's a dynamic application. And if the patient were to have side effects, you can immediately terminate it. Diamox cannot be reversed. I mean, you have to sit out 30 minutes plus minus of vasodilatation. So again, the effectiveness of using this imaging modality with a vasoactive stimulus is really to create a brain stress test. You don't want to measure baseline perfusion. I don't think it will catch all patients you're interested in. You really need to challenge the vasculature, increase the blood flow where possible to really look at those patients that have failed autoregulation, so impaired cerebrovascular reserve capacity. Resting state perfusion analysis. I know the group from Hansen shown very intriguing papers where they use kind of resting state bolts as a surrogate for cerebrovascular reactivity. This could be an alternative, but I'm still a firm believer that you need a vasoactive stimulus to really identify those patients really at risk for hemodynamic failure. So we use bolt. The reasons for that already historically for us was it's a single sequence. We can, if we want in 5 minutes, do a dynamic sequence. So apply a CO2 stimulus during this 5 minutes, and then really measure CVR. And we don't need a baseline scan in that per se. We can cover the whole brain in 5 minutes. We do 3 millimeter isotropic voxels. We can even go more down now. It's like the newer sequences, but at some points when we started 10 years, we had to fix the protocol, so we stuck to it. Also for the longitudinal data acquisitions, obviously the follow-ups. And what I also like is that the bolt contrast is basically available in every MR system. I mean, you can, even in smaller hospitals, you can scan bolts, and you don't need to really modify the sequence. So the disadvantages, well, needless to say for this crowd, but I think here we would already be quite happy that we have kind of good reading. But as you see, it's very noisy, obviously. It's very hard to really filter out a pathophysiological response. There's a lot of complexities in the methodology. That's why a lot of work obviously goes into a bolt analysis, understanding it. To be very frank, I think nobody of us understands it 100%. We have to make some pragmatic considerations and assumptions to even make it clinical feasible. And what I usually get back when I send the paper in, obviously, reviewer usually said, yeah, but you're not really measuring blood flow. I mean, obviously that's true. Hemodynamics also questionable. I mean, it's definitely a big metabolic component. So that's why I think the stress test is also good, because they have a delta. You really look at like the reserve capacity. And this is something more of a mindset, I guess, than the methodological improvement. So what we do, we use the Respret, the latest generation now, generation 4. So the patient has a mask on in the MR, which is already a challenge by itself. So you have to instruct patients as well, especially if you do that now on a routine basis. We applied quite a short stimulus. This is still an old protocol, actually from Toronto, so I could have put our protocol in, but we don't do the hypocapnia anymore. We also not go up with our CO2 in a kind of like stepwise fashion. We actually do a block. So we have a baseline, go up 10 millimeters of mercury of CO2 from the baseline, the resting state CO2 value of that person for 80 seconds, 80, and go back to baseline for another 3 minutes. This is basically how we then derived the percent bolt change per millimeter CO2 change. And as you see, because it's such a robust targeted CO2 trace with the Respret, the bolt seal kind of follows it very nicely. And it also allows us to make high resolution maps. So this is not only smoothing, this is also, I think, the quality of the data. So what other, let's say, advantages of this combination of bolts with Respret, because the Respret can really apply the same stimulus also for follow-up scans. You can really precisely target it. That's why it's quite reproducible. It shows quite high reproducibility. So this is the same protocol. It was published by the Toronto group. I think when the Toronto group reached about 600 or 700 scans, they also did this safety, tolerability, and technical feasibility study. So using really the Respret with bolts, and showed that it's actually very well to be done. And also that's why also motivating to put it in a clinical practice. It's all based on a CO2 stimulus of 80 seconds. And what we do then, this is an example of Zurich. We also present this to the physicians. We actually do a whole brain. So we really send this movie out. You can look at the CVR and the cerebellum. Brainstem, arguably, I think with bolts, I wouldn't be too adamant about that, but definitely for the neocortex, for the supratentorial brain, you can very nicely, voxel-wise, do an analysis and present these bolt CVR maps. This patient has a clear example of steal phenomenon or severely impaired CVR in the right hemisphere. So how to clinically translate it then? So as I said, the journey that we did in Zurich took a couple of years, and the privilege we have or had in Zurich is that they have water pads for to look for patients for hemodynamic impairment. So it's a PET sensor integrated in our hospital where they do Dymox challenge water pads scans. This will be the classic readout. You have this baseline scan, you apply Dymox and do a second scan with the radioactive water tracer. This would be a patient typically with hemodynamic failure stage 2. So blue brain on bold as well, I guess. This is in stage 1 already impaired. You see the more severe impairment after Diamox application. We compared these patients direct with our own technique. So we did a scan, water PET scan, and within 2 to 3 weeks, every patient also received a bold CBR. I mean, this was the most competitive time we could get, and it also shows how hard it is to get a PET even scheduled. This is a very exclusive technique nowadays. This showed actually very good agreement. So this is the trace, this is the AUC for PET, for hemodynamic failure stage 2. This is bold CBR, so the performance was definitely comparable. It's the Blend-Altman agreement. So there's also made us believe that we could at least use bold CBR as a surrogate to screen for these patients with hemodynamic failure stage 2, so with the most severe hemodynamic failure, potentially, theoretically, at highest risk for stroke. However, we haven't proven that with this study. So we went further. This is a study that just got accepted. It will be online soon on the Journal of the American Heart Association. We further quantified this bold CBR with PET. So we had 2 PET specialists do a qualitative assessment of hemodynamic failure in the 57 patients. So 114 hemispheres to really classify them as having no hemodynamic failure, so hemodynamic failure 0 basically, hemodynamic failure stage 1 and failure stage 2. And then, mind you, this is not machine learning, but we did a volume under the surface 3-dimensional accuracy index analysis and split the data into 80% training data and 20% test data, did 10,000 repetitions. And with that, we could determine cutoff values for hemodynamic failure 1 versus 2 and 0 versus 1. So really find the cutoff value when a patient would exhibit hemodynamic failure 1 or hemodynamic failure 2, because this cutoff will also allow us then to calculate the stroke risk, right? So this came back from this analysis, and with this cutoff, we came to 0.1. So the 0.1 is percent bold change per millimeter mercury of CO2. And this is if a patient has less than 0.1 on the ipsilateral hemisphere of the pathology, we would classify this patient as having hemodynamic failure stage 2. And using this, and interestingly, if you look at the literature, a lot of other validation studies, not per se on bold, usually use 2 standard deviations from a normal cohort. If we were to do it in our own cohort, we have also a large cohort of healthy subjects that have the exact same protocol with bold CBR. We would also get to this cutoff point. So it's kind of like an indirect confirmation that at least there is some truth in this specific number. Again, cutoffs are always a bit tricky, but nevertheless, we wanted to validate it. So we looked at our database. This is a retrospective analysis. We identified 130 patients that had symptomatic steno-occlusive disease, so atherosclerotic disease, no MOIAs, they were excluded from this group of the anterior circulation. And we used the threshold, as I said, corrected for time between stroke and the bold CBR scans. Sometimes the time was a bit large, a couple of weeks, age, sex, but also the presence of atrial fibrillation, which was the strongest control. And interestingly enough, what we saw there is that 28 of these 130 patients over time, so after the bold scan, where they showed this bold, they showed recurrent ischemic events. So the median time was 85 days. And obviously, this is the most interesting part, because the patients that were grouped in this hemodynamic failure stage 2 group, so patients with bold less than 0.1, so our cutoff for hemodynamic failure stage 2, those were, in those group, 44 patients had this recurrent ischemic event. Patients that had, let's say, not impaired CBRs or larger than 0.1, only 7%. And if we then compare it in this in a regression model, this actually showed that if these patients have this cutoff, so value of bold CBR below 0.1, they have a 10 times higher risk for recurrent ischemic events. Another interesting finding is that this 44% corresponds very well to the literature I just showed you. This is a lot of studies that were done before and kind of come to the same conclusion that in these patients, so symptomatic patients with atherosclerotic large vessel disease and hemodynamic impairment have the highest risk for stroke. So we kind of indirectly confirmed it independently with our own analysis as well. So this is the Kaplan-Meier curve of these findings. And what you see, this is the group with the bold CBR cutoff below 0.1. This is the patient of 0.1, so no hemodynamic impairment and hemodynamic impairment. And also from the time distribution, this also fits very well with other literature. Most recurrent stroke really happen in the first. Again, if you have the hemodynamic impairment, at least in all cohorts, using bolt CPR, it's 10 times higher risk of recurrent ischemic events. And this is important data because this we can now integrate in our clinical decision making. It doesn't mean that patients need a bypass. We've seen many bypass trials now that didn't show efficacy to reduce recurrent stroke risk. However, we have strong data now to really say, okay, but maybe we have to do some kind of therapy on these patients. We cannot leave them because the stroke risk is significantly increased. So to move to the acute setting, again, whole different beast. I mean, to even integrate advanced hemodynamic imaging, needless to say to everyone, it's very hard to already put in the clinics. If we don't then talk about acute ischemic stroke patients that are acutely, obviously suffering from symptoms, need to be hospitalized, and then also try to impose an MRI on them, even from the infrastructure, it's quite challenging. So we were a bit lucky that with our neuro clinics, meaning stroke neurology, cerebrovascular neurosurgery, and interventional neuroradiology, we got a grant from the university about 4 years ago that explicitly funded this, this project to really not interfere with the clinical management. So the patient comes in, we don't do anything. The patient gets the normal stroke workup, gets presented to the stroke neurologist, get a CT perfusion, endovascular thrombectomy, yes, no. It's just that patients after that would get a hemodynamic imaging protocol. This included MR perfusion, baseline perfusion. So the standard, I would say, for patients that remain symptomatic, but also bolt CPR within 72 hours after the thrombectomy. So that was kind of the cutoff, easily set from this project. And with that, we had the possibility to restudy two phenomena. So what we wanted to do is really look at steal phenomenon in these patients. So this negative CPR, basically hemodynamic failure stage 2. If you want to use a... So patients came in, got the clinical workup and evaluation, as I said. Patients that were eligible for endovascular thrombectomy using the current guidelines got thrombectomy. What we were interested in were those patients that had successful recanalization, meaning occlusion was successfully reopened. So the vessel was opened. However, these patients remained symptomatic and showed reperfusion failure, so really hemodynamic failure on this advanced imaging. It's also a group we don't standardly identify, right? I mean, it's not a standard workup. The other group that was of interest to us were those patients that were considered ineligible for endovascular thrombectomy under the current guidelines, but still showed neurological deterioration during hospital admission. It's basically an exclusion group nowadays in most hospitals, and we still don't really know why they still have symptoms. So also here, we want to investigate whether the hemodynamic impairment could be present or could explain the neurological deterioration. So also here, these people got within the 72 hours this bolt CPR and MR perfusion scan. The first project for the reperfusion failure, we scanned 30 patients, but had to exclude 9 of them for didn't have the bolt CPR data at the end, meaning the scan was either not done well or we sometimes they clean up the scanner in our institution, but like Swiss style, every 24 hours. So if you don't download your data, it's gone. It's a bit of a shame, so it's a bit painful to even show it. 5 patients exhibited excessive head movement. This is something we know and we kind of calculate, although we try to fix the head. But again, acute stroke, people are not per se that happy to do an MRI, right? So 21 patients, I thought was quite good. These are the demographics, and I think what's most interesting is if we focus on these 2 plots, because this shows the bolt steal volume. This is the DWI, so the stroke volume. And what we show is that the larger the stroke volume, the larger the steal volume would be, but not per se on the DWI lesion. So we didn't see a spatial agreement per se. It just shows that the larger the DWI, so the initial stroke, the larger the steal volume somewhere in the brain of this patient was based on bolt CPR. So below this cutoff of 0.1. This is the NIHSS discharge, so the clinical performance basically, versus the steal phenomenon. And what we showed here is that the larger the steal volume present in these patients, the higher the NIHSS, so the worse the clinical outcome at discharge was. So these are 4 examples, patient A, B, C, and D. This is the bolt CPR map. This is the bolt associated steal phenomenon. So what we also do now for the clinicians, because this is also for us, I mean, for a trained eye, we already see where the impairment would be, but we kind of highlight steal. So the negative steal changes, we also present the steal map. And for this paper, we also highlighted the steal in these patients. And this is the DWI volume. And what you can appreciate is what we show a bit here, is that this spatial agreement obviously is only partially of the DWI lesions. So we see a lot of steal outside the DWI lesions, or no steal where the DWI lesion is. This could be a reversible phenomenon, right? We also know that DWI lesions may be reversible over time after successful thrombectomy. This is all done at baseline, obviously. This is done within 72 hours, so that also may explain a bit of the mismatch. This was just published last month. So again, all the data I'm showing is either not published or really published this year to kind of show the newest findings we have. So now this whole reperfusion, what is really relating this? Why would we ever see steal in patients that have an reopened vessel, right? So there's a lot of theories. Obviously, at this point, we still don't know. I think a very nice review that just came out September by the Columbia group kind of postulates certain explanations, but at the end, it will come down to a macrovascular or microvascular dysfunction that's really that gives this persisting hemodynamic failure. So I think what we show with BOLD now, preliminary although, is that there is actually hemodynamic failure that was also not known, and this may also be related to the insufficient capillary reflow, the microvascular occlusion, microclots. It's even inflammation theory. So at the end, a lot of room for thoughts and interpretation. One study I wanted to highlight in this regard, and showed a very interesting explanation for this reperfusion failure, is that potentially an early venous filling, so collateral activation or inactivation, may explain part of this persisting hemodynamic failure, despite the vessel being open. This is done by a stroke neurologist. We were not involved, but what they showed is that they studied the leptomeningeal collateralization. They used a rodent model with larger vessel occlusion stroke. They also found in this model this steal, so persisting steal. It's kind of confirmed our clinical finding. What they did then, because they also found the impaired reactivity, they then looked at 33 clinical patients as a side project in this paper, and then looked at those patients that got thrombectomy done. So they got a diagnostic catheter angiography, and then looked after the vessel was open, did another contrast run, and then really looked at after the thrombectomy and the DSA, it was early venous filling. So if the brain would, the sagittal sinus would fill early, that would be they interpreted that would have that would correlate to a lower collateral status before thrombectomy, and also a higher risk of hemorrhagic transformation of the stroke that was present. So I thought it was an interesting finding, and I mean it's a very large leap. But if we talk about sickle cell as well, about the shunting lesions, I think there's a lot of thought in stroke also about collateral activation. So the collaterals are there, they may be inactivated, so they may, in this case, may not work as well. So the venous, the blood shunts into the venous system, or they may be active and may therefore prevent a larger stroke. So I think, again, a lot of room for thought, but the data they showed in this paper is still not published. It's on Matt Rivic's. That's early venous filling. So after thrombectomy, if you then do another contrast run, that could show another imaging correlate for reperfusion failure. So what we would like to do now is to correlate our BOLD CBR findings, really look at steal phenomenon patterns, and look at the early venous filling on the DSA as the next project. So now looking at those patients that are coming in, acute stroke, on the current guidelines, do not get the reopening of their vessel, so the vessel occlusion persists. And we looked at that group that did not move or stay the same, but really deteriorated. So they showed a neurological worsening during hospital admission. Then look at those patients. I just show you some box whisker plots here. At the end, this analysis is done on 43 patients. These are BOLD CBR values of the whole brain. And the neurological deterioration was either no or yes. So the patients that did not deteriorate had higher BOLD CBR values. Still in the impaired range, it's still 0.1 till 0. I mean, if we look at the distribution, but really if they were deteriorating, we also see a CBR effect, so really a hemodynamic effect correlating to the neurological deterioration. This plot shows the steel volume in the brain. And again, neurological deterioration, yes or no. So with patients that did not have deterioration, stayed clinically stable or improved, there's less steel present in the brain. Obviously, interesting is those patients that had larger steel. These are not an insignificant amount of patients. 7 patients showed large steel phenomenon, but did not deteriorate. So, I mean, obviously, this is something we can only speculate about, but it would be an interesting group also to look. And this may also, if you're very negative, say, well, maybe the Bolt CBR does not show any hemodynamic failure. But again, these are the outliers. The last box whisker shows the penumbra core mismatch. So these are the patients that would typically, let's say in other institutions, would get an MR perfusion. These are patients that get the advanced imaging with baseline perfusion. And if we look at the baseline perfusion, so this would be the penumbra core mismatch volume, deterioration no versus yes, there was no significant difference. So if we were to scan these patients with our standard baseline perfusion measurements, we may miss those patients that are actually deteriorating and have a hemodynamic underlying problem. And I show this in this last slide with 4 examples. I orientate you a bit, but these are again 4 patients, DWI, Tmax, over 6, so the standard cutoff we would use now internationally to say this is a mismatch, or at least like hemodynamic impairment. These are different cutoffs. Blue is less than 4 seconds, up to red more than 10 seconds. And this is the Bolt CBR. So the Bolt CBR in this patient and the steel volume, again, isolated from the Bolt CBR. So this is a typical patient, has an occlusion, one-sided, ICA occlusion, small stroke, large mismatch, right? So even on MR perfusion, we already see a large mismatch, very correlating very well to Bolt CBR. So this is a patient not eligible for thrombectomy, but still has a large hemodynamic deficit, although he has a small, so big mismatch because has a small stroke. This is a patient we nowadays, study-wise in the study design, do an acute flow augmentation bypass. So this is a patient in Zurich that would get a flow augmentation bypass surgery. I'll show you one example after, because otherwise we would not have a treatment option for this patient to restore blood flow to the brain. Yeah, sorry, yeah, I'm almost there. So thanks. This will be a similar patient, no stroke, big mismatch again, steel. Here we'd also entertain a bypass. Patients that are more interested from an academic or scientific point of view are also patients with little stroke, no mismatch, but a large Bolt steel volume. So are we really looking at hemodynamic impairment here? Are we missing it here on MR perfusion due to technical limitations to the 6 seconds cutoff? So these are patients that are very interested to also longitudinally follow up, because if they were to make a stroke, we may be more confident that it's really related to a hemodynamic problem, right? Or at least they may be at high risk for recurrent stroke in this area. So this are patients we would now novelly be able to identify with Bolt CBR. We would not typically pick up on our standard imaging for acute stroke at this point. Another patient, also no stroke, no significant mismatch. So this over 6 seconds Tmax is a Tmax in the 4, 6, 8, and 10 range. So blue would be 104 seconds. So arguably hemodynamic impairment, but according to guidelines not. However, we do not, so we would typically say no hemodynamic impairment in this patient, and we do see a small steel volume. This would also be interesting in these patients to kind of correlate MR perfusion with Bolt CBR, but also look whether there would be a stroke, or at least the patient would clinically deteriorate. So just some examples from very preliminary data where Bolt is an advanced hemodynamic imaging technique, may add novel, but well, may be a novel parameter to also take into account to study these patients. So again, for the patients, that's quite obvious where they have a little stroke and large mismatch. What we do now is that they get a bypass, not the indirect bypass, but really a direct anastomosis, meaning it's quite a small surgery. So make a small craniotomy, use the superficial temporal artery, like the classical flow augmentation bypass graft, and then suture it onto one of the cortical middle cerebral artery. And then it is usually typically carries around 30 or 40 milliliters of blood flow. What we do see, and that's quite dramatic, and I think that's why the etiology in acute stroke is completely different than chronic stroke, is that patients obviously have a lot to lose because the occlusion is acute, the hemodynamic failure is acute, And even if they have a small stroke, they may still lose out a lot on their brain tissue. So if we were to operate these patients, what we really see is a dramatic improvement. Clinically, within days, these patients typically get much better, and also the bolts, even after weeks, already show almost normalization. This is what we typically don't see in chronic patients, and that's also obviously where we don't see the efficacy of bypass per se in atherosclerotic patients with more chronic acute stroke. Although experimental at this point, I think it's still worth investigating, at least to have another therapy for these patients that are excluded nowadays, or still excluded for endovascular thrombectomy. So to wrap it up, so Bolt CPR can definitely be integrated as an advanced hemodynamic technique for patients with symptoms derived from atherosclerotic large vessel disease and chronic ischemia. So these patients that typically already have had a stroke and are known in the clinic. If we study this patient, we see actually a tenfold increased stroke risk if they have this Bolt CPR cutoff value below 0.1% bolt change per millimeter CO2. And if we take Bolt CPR into acute ischemia patients, we may be able to use it as a novel or an add-on, I have to say, add-on advanced hemodynamic imaging technique to study reperfusion failure, study mismatch versus MR perfusion, baseline perfusion versus Bolt CPR, and maybe better characterize these patients that are currently not operated on, that cannot be revascularized, current guidelines or techniques, but still deteriorate during hospital admission, to really understand there's a hemodynamic component to this etiology. So with that, many thanks to my team. I have two very motivated postdocs, Bas and Martina. They also became neurosurgeons, they finished their PhD in my lab. So kind of like the same profile, and two aspiring neurosurgeons that are currently running the show. And so thank you very much.